Darwin's Origin of the Species. And I quote, he wrote, "All living things have much in common in their chemical composition, their germinal vesicles, their cellular structure, their laws of growth and reproduction. Therefore, I should infer from analogy that probably all the organic beings which have ever lived on this earth have descended from one primordial form into which life was first breathed." Clearly, Darwin's view of descent by modification led him to the conclusion that there existed sometime in the very remote past an organism that is literally the universal common ancestor of us all. This is the final lecture in the first section of our course, which is focused on the theme of information and evolution in living systems. I began this section of the course with the question of how life could have arisen from non-living matter. At some point in the remote past, this life was, as Darwin put it, somehow first breathed into a primordial form. An entity appeared that we would all agree was living. Once this entity had gained a foothold on life, it became, in a very real way, the common ancestor to everything that has ever lived on the planet. In today's lecture, I want to go back and take a look at this theme of information and evolution by considering that first organism and tracing some of the major milestones in the early history of life that must have followed its origination. Of course, I'll only be able to do this with the very broadest brushstrokes, but I think this is worth doing because it gives us a different sense of the enormity of evolutionary history by looking at how the fundamental pieces of the puzzle that had to be established early on were set up. And in that sense, it provides a useful capstone to everything we've discussed up to this point. Exactly when this first living organism arose is uncertain, but we do know a few things. We know that it has to be sometime after about 3.9 billion years ago, when the surface of the Earth was no longer being bombarded by stellar debris and the crust of the planet had cooled sufficiently for liquid water to appear. And it had to be sometime before 3.5 billion years ago, which is the date of the oldest fossil cells that have been found so far. These oldest fossils are found in rocks in Western Australia. They're actually quite advanced in their appearance, resembling certain kinds of modern bacteria, so most biologists assume that the first cells must have appeared even earlier than this, perhaps several hundred million years earlier. Let me remind you of some of the characteristics of this earliest living thing. This first organism was certainly a single cell. It had to have a membrane that created a compartment separating it from the rest of the world, allowing the accumulation of specialized organic molecules within. Among the organic compounds that made up this first organism, there must have been complex polymers capable of catalyzing biochemical reactions, the kinds of reactions that would be necessary for this primitive cell to process materials, grow, and divide. The most special and specialized of these biological polymers must have been some kind of molecule that could not only catalyze reactions, but also serve as a template for making more of itself. In the earliest cells, as we discussed early on, this information molecule probably was some form of RNA. Later, after a primitive genetic system was established, in an RNA world, DNA took over the role of storing and transmitting genetic information, while proteins took over the principal role as the functional molecules responsible for everything else the cell might do. No doubt, at this early period in time, the major effect of natural selection on this primitive organism was to refine its genetic system and to somehow ensure that the different kinds of molecules within it would cooperate. There had to be some selection so that these different kinds of molecules, the lipids, the proteins, the nucleic acids and sugars, would be able to specialize to do their own jobs in coordination with each other. Now, by the time we get to the oldest fossils, about 3.5 billion years ago, evolution had established a recognizable cell, a prokaryotic cell, which is, as far as we can tell, not too different from what might be a typical bacteria we find today. There's actually quite a rich fossil record back then, and a fossil record that continues from that period up to the present day. And this fossil record makes a couple of things clear. First of all, it shows that when life did emerge, it emerged very rapidly and quickly filled the planet. Life was really teeming in the early Earth, even if it was only single cells. Second, it also shows that only prokaryotic cells existed from those earliest cells for about another 1.5 billion years, until about 2 billion years ago. In other words, that more complex kind of cell, eukaryotic cells, didn't exist for quite some time, and the early evolution of life on this planet was all in the context of prokaryotic cells. Now, the fact that prokaryotic cells essentially ruled the Earth for 1.5 billion years might sound boring, but it's nothing of the sort. There were at least two major evolutionary events that occurred during this period. The first was actually that the prokaryotes themselves split into two major lineages of kinds of cells. It's interesting, when I was a student, this wasn't recognized. We thought prokaryotes were prokaryotes and eukaryotes were eukaryotes, and there were no distinctions within them other than species differences that arose later on. But today we recognize two kinds of prokaryotes. We call these, on the one hand, the bacteria, and on the other hand, the archaea, which comes from the Greek root for ancient. The differences between bacteria and archaea are relatively subtle in the modern world today. There are biochemical differences in the outer coatings of the cells, in their internal biochemical composition, and so forth. And in fact, there are enormously more bacteria in the world today than there are archaea. The archaea largely have been relegated to very extreme environments. These are the prokaryotes, for example, that we find in hot sulfur springs or in extremely saline water, such as that found in the Great Salt Lake. So, in a sense, archaea are not necessarily an important form of life for us to understand today, but as we'll see in a moment, they're actually very important for understanding the origin of eukaryotic cells, from which were derived from them, but in a very complicated way. Now, the other thing that had to be happening during this period was the evolution of an enormous number of different kinds of biochemical pathways that we would today call metabolic pathways. Biochemical pathways that were used to obtain energy, process food, build macromolecules, and otherwise carry out the biochemical functioning of the cell. It's clear some of the biochemical pathways which are still central to the functioning of cells today arose very early on. For example, glycolysis refers to a metabolic pathway which breaks down sugar molecules. We'll look at this in more detail in the third part of the course, this and other metabolic pathways. In so doing, glycolysis takes energy that's stored in those sugar molecules and makes it available for the cell's use. As prokaryotes multiplied and diversified, competition for limiting resources must have led to the evolution of increasingly diverse ways to acquire materials from the outside, or in other words, increasingly diverse metabolic pathways. So the early evolutionary history of prokaryotes is really the evolution of a variety of metabolic pathways, which in turn really is the evolution of proteins, because all of these metabolic functions are mediated by protein enzymes, which are what are catalyzing those biochemical reactions. Of course, this kind of evolution of proteins and metabolism isn't something we can observe in the fossil record, but it is something we can deduce from the kind of phylogenetic analysis we talked about last time when we look at the structure of macromolecules and the DNA that codes for them. It's clear that nearly all of the metabolic processes that we find in modern cells today evolved in prokaryotes before eukaryotes ever appeared on the scene. Now, the evolution of one biochemical process, one metabolic pathway in particular, had an overwhelming effect on the subsequent history of all life on Earth. Because early prokaryotes must have been competing for organic resources that would provide energy, then clearly the evolution of a process that could tap into a new, boundless source of energy would have been a tremendously successful adaptation. One such source of energy is sunlight. As we'll learn in detail later, photosynthesis is a biochemical process by which the energy of sunlight can be captured and used to build sugars, which in turn store that energy for the cell until it releases it through glycolysis or other kinds of metabolic pathways. Molecular phylogenetic data suggests that photosynthesis also arose very early in the evolutionary history of prokaryotes. And because of the obvious selective advantage of this trait, those organisms that possessed it soon became a dominant force on the planet. Now, there are several different kinds of photosynthetic pathways existing in prokaryotes today. The most efficient kind of photosynthesis, which is found in modern plants and also in a group of bacteria called cyanobacteria, has an interesting property. Its efficiency is coupled with the fact that it happens to generate oxygen as a waste product. Thus, beginning with the evolution of photosynthetic pathways, oxygen began to be produced, and produced in abundant quantities. Now, the fossil record and other evidence suggests that these cyanobacteria probably appeared around or before about 2.7 billion years ago. Therefore, beginning about that time and continuing for some time after, generation of oxygen increased and increased as this photosynthetic adaptation increased, such that within about 1 billion years or so, the entire atmosphere of the planet Earth had been transformed from one which had no oxygen in it to one which, like today, is composed of about 20% oxygen. Now, what's the significance of this? The significance is that oxygen is a highly reactive molecule that interacts with organic molecules, breaking them down, oxidizing them, as we say. And in fact, the presence of oxygen proved to be an environmental disaster of global proportions for most of the organisms that lived before oxygen appeared on the scene, because they simply could not live and function in an oxygen-rich environment. Remember, this was the sort of key to the insight to how organic molecules could evolve in the first place. The Haldane-Oparin theory that Miller tested in that famous experiment when he first synthesized organic molecules assumed that the atmosphere had to not have oxygen. And when oxygen appeared on the scene, that kind of spontaneous synthesis of organic molecules could no longer occur. And furthermore, those organisms that had evolved in a non-oxygen environment now were in very sorry straits. The biochemical and physical adaptations of organisms that had accumulated up to that time couldn't cope with the oxygen revolution. But a few kinds of organisms did evolve some mechanisms that allowed them not only to cope with it, but in fact to take advantage of it. And specifically, some organisms developed a completely different process for metabolizing energy-bearing molecules like sugars, a process that we call today cellular respiration. The metabolic pathways of cellular respiration actually take advantage of the presence of oxygen to enormously increase the efficiency with which energy can be extracted from organic molecules. Thus, the evolution of photosynthetic pathways radically and permanently changed the Earth's atmosphere and the biological inhabitants living on it. But in the long run, it made certain changes that were key milestones in the history of life on Earth. Notably, it increased the amount of energy that could be produced by cells in two ways. In essence, cells had found both a new fuel, that is the sunlight that they could gain access to through photosynthesis, and a better way to burn the old kind of fuel, that is the metabolic pathways associated with cellular respiration. This increase in the amount of energy available permitted cells and organisms to become ever more larger and complex. Now, another positive consequence, I should add, of the accumulation of oxygen, something that didn't happen for quite some time later, actually 1 billion or so years after the oxygen atmosphere began to accumulate, was the development of a layer of ozone gas in the upper atmosphere of the Earth. Whereas oxygen had a negative effect on the organisms living on the planet at the time that it first accumulated, ozone actually had, in a sense, a positive effect, because ozone absorbs the radiation that hits the planet, and in so doing made it possible for organisms to inhabit environments that previously were unavailable to them, most notably leaving the water and entering a land environment. Now, at some time, probably actually not long after photosynthesis and cellular respiration evolved, organisms started to eat each other as well, eating each other whole. Of course, single cells don't have mouths, so the only way that one cell might eat another is by physically engulfing it, literally wrapping its cell membrane around the other cell until the prey cell is completely surrounded and engulfed on all sides by the cell body of the predator cell. As prokaryotes diversified and competition for limiting resources increased, it makes sense that some cells would want to start eating each other this way, because to do so would be a very efficient way to obtain a big package of organic molecules all at once. Odd as it seems, the evolution of eukaryotic cells in specific may be largely the consequence of cells trying to eat each other and getting indigestion. This idea is known as the endosymbiotic theory of eukaryotic evolution, and it was first proposed by Lynn Margulis of the University of Massachusetts at Amherst over 30 years ago. The endosymbiotic theory suggests that at least two of the most important organelles found in eukaryotic cells originated when one prokaryote engulfed another, and instead of ingesting it, instead of breaking it down, developed a symbiotic relationship with it. The Greek root symbio means living together. Endo means inside, hence the endosymbiotic theory of eukaryotic evolution. Now, to get to endosymbiosis in this way, probably the first thing that had to happen was that the cell membrane of the original eukaryotic cells had to evolve to be more flexible. Once a cell has a flexible membrane, it will be able to fold its membrane around and engulf another cell, but it will also be able to do other things, specifically invaginate in on itself so that it can fold off and pinch and make compartments that are internal compartments. So, for example, an infolding of the cell membrane is thought to have given rise to the nucleus of modern cells by providing a membrane-bound internal compartment in which the DNA of the cell could be sequestered, and by being sequestered, protected from other biochemical activities of the cell. As you know, the genomes of eukaryotic cells are much larger and more complex in their organization than those of prokaryotic cells. It's not clear exactly how the origin of the nucleus is related to the evolution of increased size and complexity of the eukaryotic genome, but there's little doubt that the existence of the nucleus in modern eukaryotic cells is absolutely essential for the ways in which these cells manage their DNA, not only on the molecular level, but also on the cellular level when cells undergo mitotic or meiotic cell division. Well, now let's get back more specifically to the endosymbiotic theory. Once cells had developed a flexible cell membrane, larger cells might begin to engulf smaller cells, literally as a way to obtain resources, as I said. Lynn Margulis contended that it was possible that a small prokaryote occasionally became engulfed, but failed to be broken down. The smaller engulfed cell would now be trapped inside the larger cell. Of course, this arrangement could only be maintained over time if both cells, the smaller one trapped inside and the larger one that had trapped it, benefited in some way from the arrangement. In other words, that this arrangement was adaptive in some way to both cells. The advantage to the cell that had been engulfed seems obvious. It's now living inside of a very nutrient-rich environment, much more so than if it was outside in the real world. But what could be the advantage of to the large cell now serving as a host to the smaller cell that it engulfed? Margulis argued that there would be an advantage if the cell that was engulfed happened to have a more adaptive set of a metabolic pathway, rather than the predatory cell that had engulfed it. And there are two such pathways that had evolved, remember. Those are photosynthesis and cellular respiration. So specifically, Margulis suggested that the evolutionary origin of the organelles called mitochondria, which are specialized for energy processing, as we'll learn later in the course, occurred when a cell capable of cellular respiration became endosymbiotic with a larger cell that lacked these pathways, that could only perform glycolysis. Remember that cellular respiration is much more efficient in the way it extracts energy from the breakdown of organic molecules. These protomitochondria, the engulfed cells, would benefit by having a buffer from the rest of the world and a constant supply of nutrients provided by the larger cell that it was within. In return, the engulfing cell, the larger cell, gained energy that was produced by the cell it engulfed. So if the smaller cell was able to allow some of the energy it produced to go to the larger cell, then it would be like a little energy-producing factory, which is exactly the way that mitochondria work today. Similarly, Margulis suggested that chloroplasts, these are the organelles that are specialized to perform photosynthesis in modern cells. These are the organelles that are specialized to perform photosynthesis in modern cells. That chloroplast originated when an early eukaryotic cell engulfed a smaller photosynthetic cyanobacteria. As natural selection acted to make the host cell and its endosymbionts more interdependent, this confederation of cells, cells that are living together in a symbiotic relationship, would eventually be integrated into a single organism, the single organism that was a eukaryotic cell. Now, Margulis's endosymbiotic theory was greeted with skepticism when she first proposed it. But over the decades that have followed, a growing body of evidence has accumulated to suggest that she had it exactly right, that something like this happened maybe 1.5 billion years ago. Much of the original evidence was circumstantial. For example, if you look at the structure of mitochondria or chloroplasts, you see that they have not one membrane surrounding them, but two. And this is what you would predict. If a cell had been engulfed and maintained its own membrane, but was surrounded by the membrane of the engulfing cell, you would predict two membranes. There's also the interesting fact that mitochondria and chloroplasts have their own genomes. They have their own complement of genes. Now, it turns out not only do they have their own genomes, but these organelles replicate by cell division themselves. So what this actually means is that when a eukaryotic cell divides, such as by the process of mitosis that we talked about before, in advance of that, the mitochondria and the chloroplasts, this is a chloroplast-containing cell, themselves have to divide so that when the cell divides, when the eukaryotic cell divides, there's enough chloroplasts and mitochondria to go around. The eukaryotic cell itself doesn't replicate these mitochondria and chloroplasts. They replicate themselves. And then they divide themselves, or they are divided, into the daughter cells of a mitotic division. The most interesting piece of evidence, by the way, that suggests that mitochondria and chloroplasts actually are ancestors of endosymbionts in this way has come from phylogenetic studies of the mitochondrial and chloroplast gene sequences themselves. If you look at the structure of the gene sequences found in these organelles, you find that they resemble, from a phylogenetic evolutionary perspective, their presumed ancestors, the cyanobacteria or other bacteria from which lineages they were thought to have originally been derived. One other interesting thing about these mitochondria and chloroplasts is that they have evolved together with the cell, the eukaryotic cell, again going beyond a confederation into one integrated organism, to the extent that although they carry their own genes, they don't carry enough genes to live entirely on their own. So the genetic function of a mitochondria is in part due to genes that it bears in its own genome, but in part due to genes that are found in the eukaryotic cell's genome found in the nucleus. So it is a true melding of genomes from these two sets of cells. Now, the last interesting twist in our understanding of the evolution of eukaryotic cells is to ask, who were the progenitors here? It turns out that genetic evidence suggests that they were the archaea, not the bacteria, that gave rise to the engulfing cell, to the larger cell that became the eukaryotic cell. But the organelles that were engulfed were bacteria. So what that means is that after the initial division of life into two major lineages, the archaea and bacteria, there was a reintegration of those cells to form this amalgamation, or really a true chimera, as an innovative complex cell by joining these major cell lineages? Well, in a single lecture, of course, one can't do justice to the extraordinary grand scope of all evolutionary history. So we have to accelerate our tour a bit. The evolution of eukaryotic cells is thought to have occurred some 2 billion years ago or so. This led to an enormous diversification and innovation, and the pace of evolutionary change quickened. Eukaryotic cells diversified into a broad range of single-celled organisms, organisms that we call protists today, and which continue to be abundant and ecologically important. More strikingly, however, some lineages of eukaryotic cells gave rise to true multicellular organisms. Now, the evidence for when multicellular organisms first appeared is conflicting. If we look at the molecular phylogenetic studies, they suggest that the first eukaryotic multicellular organism might have lived as long as 1.5 billion years ago. The oldest fossil that we could conceivably argue is truly multicellular is about 1.2 billion years old. But even more convincing fossils don't really appear until as recently as 600 million years ago or so. So when multicellularity arose isn't quite clear, but the significance of multicellularity is much more clear. This condition enormously expanded the potential for evolutionary diversification that living organisms could explore. It did so for a number of reasons. First, now a single organism could be composed of many cells, each of which could specialize for a single function. In other words, selection could act in different ways on different cells so that they could become more specialized. There would be fewer trade-offs in selection, and cells could do just one thing very well within the amalgamation of the multicellular organism as a whole. The evolution of multicellularity also allowed organisms to become larger, much larger. Through the first couple of billion years of evolutionary history, there are few, if any, organisms that you can see without a microscope. But once multicellularity evolved, organisms could become very large. And in doing so, not only could they become large, complex things like the, say, elephants and blue whales that we see today, but more significantly and more fundamentally, they could create an internal environment. An environment within the amalgamation of cells that allowed cells on the inside not only to specialize, but to exist in an environment, the internally created environment, that was much more favorable than the harsh external environment that they might otherwise live in. And that allowed then the specialization of cells on the outside of a multicellular organism to deal with harsh environments, again allowing a greater range of places and ways that organisms could live. Perhaps the most significant consequence of multicellularity was that certain cells within an organism's body had to become specialized for sexual reproduction. With single-celled organisms, all we need is mitotic division. But the evolution of specialized reproductive cells was something that had to happen when we now had a number of cells with different specialized functions. Some of those cells had to be sequestered and saved specifically for the task of making more individuals. And as a correlate of that, a different kind of cell division process must have evolved. The process of meiosis, which, as we've seen now in detail, creates enormous potential for genetic diversity through recombination. And of course, with the advent of sexual reproduction and the generation of genetic variation that is a consequence of it, now there was an enormous opportunity for evolutionary change. Well, if we reduce the entire history of the planet Earth to the scale of a single calendar month, the appearance of multicellular organisms would occur somewhere around the 24th day. Most of the month has already passed by the time life has made it past single cells. From this point on, however, things accelerated very rapidly and increasingly became adapted and diversified into the untold forms that appear on the planet today. Of course, there's so much that we could talk about in this regard, but it's time for us now to move on and explore life from other perspectives. The theme around which we've built this first section of our course has been information and evolution, and we've now looked at biology across all scales of the hierarchy of life from this perspective, beginning with the problem of how a single primitive cell could store, copy, and transmit information. Where we've ended up just now, with the advent of multicellularity as life expanded and diversified, actually takes us to the theme of the second section of the course, which will begin in the next lecture. Once organisms became aggregates of cells, leading in a few hundred million more years to complex assemblages such as ourselves, a different set of problems arose. How do the different specialized cells in a multicellular organism coordinate their activities? How do some cells create an internal environment that provides protection for other specialized cells that are no longer able to completely fend for themselves? How do different types of specialized cells arise from a single-celled zygote after gametes have joined to develop a multicellular organism in the first place? These are the kinds of questions we'll address next when we begin talking about development and homeostasis, which are the twin themes of the next section of our course.